Hello, in this video we're going to talk about enzyme activity and factors that affect enzyme activity. So enzyme activity is the rate at which an enzyme catalyzes a reaction. So there's a few different ways we can measure this. One is turnover number. So that's the number of substrate molecules acted on by one enzyme molecule per minute. So if you only had one enzyme available and it had to act on a bunch of substrate molecules, the turnover number would tell us how many of those substrate molecules could be converted to product in 1 minute by that one enzyme. So it would act on one, spit it out as products. It would act on this one, spit that out as products. And the turnover number tells us how many substrate molecules the enzyme can convert. So it's kind of like if, let's say you're wrapping presents and it's just you wrapping presents, your turnover number would be how many presents you can wrap in 1 minute without any other help. So let's look at some examples of turnover numbers. So this table's in your textbook. And you can see that there's a range of turnover numbers. So for instance, carbonic anhydrase, which catalyzes the conversion of carbon dioxide and water to something called carbonic acid, its turnover number is 36 million. So that is a lot. But then if we go down, we can see chymotrypsin, which hydrolyzes specific peptide bonds, so it breaks apart certain bonds between amino acids. This can only convert or break apart bonds, 6,000 bonds per minute. Now that's still a lot, but relative to carbonic anhydrase, that doesn't seem like it's a lot. So it really does depend on the enzyme and how quickly it can function. Now, another way to measure enzyme activity is by using what's called the Enzyme International Unit. So this is the quantity of enzyme that catalyzes the conversion of 1 micromole of substrate per minute. So this would be if you had multiple enzymes available and you just wanted to see how quickly those enzymes could convert a substrate to product per minute. And you would just have a specific concentration of substrate, which is 1 micromole. Now, if you need to make a measurement like this, you would use what's called an enzyme assay. So this is the experiment that measures enzyme activity. So now let's talk about factors that affect that enzyme activity. So there's 3 main factors: enzyme concentration, substrate concentration, and the environment. So enzyme concentration would be how many enzyme molecules or proteins are available. Substrate concentration, how many substrate molecules are available or ready to be acted on. And then the environment. So this would be things like, is it in an aqueous environment? Is it in an alcohol? What is it dissolved in? And then the pH of the environment. Is it acidic? Is it basic? Is it neutral? There's also things like temperature, etc. So we'll go over all of that. But let's start with enzyme concentration. So the more enzyme that's present, the higher the enzyme concentration, and the faster that the substrate can react. So the greater the enzyme concentration here on the x-axis below, the greater the rate of the reaction, which is on the y-axis. And this is a linear relationship. So this is pretty simple. So let's say you're wrapping presents again, and it's just you initially, but then you ask some friends to help you out. The more friends you have helping you wrap presents, the more presents can be wrapped. Now let's say instead of increasing the enzyme concentration, we actually increase the substrate concentration. So this can also increase the reaction rate, um, but only up to a point. So you're, let's say you just have three friends helping you wrap presents, and you have a bunch of presents. So the rate of producing wrapped presents will increase the more presents there are to wrap. However, you only have three friends there helping you, so you're kind of limited in how many presents you can wrap per minute, right? So you do reach a saturation point at which you can only wrap so many presents per minute, right? So the graph below looks a little bit different. It starts to plateau because there's only so many presents to wrap, or so many substrates to convert to products. And then the environment. So the environment refers to things like temperature, pH, so acidic or basic or neutral. The solvent, are you in a nonpolar solvent or a polar solvent? Are you in an aqueous environment or something different? And then there's also physical movement. So let's start with temperature. So enzymes have an optimum temperature range, and that's usually between 25 to 40 degrees Celsius, above or below which they begin to denature. So an example graph is shown below. So temperature is on the x-axis and rate is on the y-axis. So as temperature is increasing, the rate of the reaction might also initially increase just because we're providing more energy with that added temperature and added heat. But eventually you're going to hit a point at which the enzyme can no longer function because it's too hot. So that's where the enzyme, the protein, could start to denature and fall apart, and then it no longer functions. So remember in our last chapter on proteins, we said that one way that you could denature a protein is with heat. Now enzymes also have an optimum pH value at which they function. So this is usually around 7, so kind of at a neutral pH. And then if you're above or below that, the rate will decrease. So let's say that our optimum pH value is 7. If you get too much below that, the rate will decrease. If you're too far above that, the rate will also decrease. You might still get some action going on between the enzyme and the substrate, but it might not be as efficient. And then at a certain pH, the enzyme will likely denature or fall apart. Okay, so let's review really quickly without looking at your notes. See if you can list the 3 main factors that affect enzyme activity. So you can pause the video, and then when you're done, we'll go over it together. Okay, so let's list those 3 main factors. So one of them was enzyme concentration. So remember, the rate of the reaction will increase the more enzymes that are available to help with the reaction. The next one was substrate concentration. So remember, the goal here is to produce product. So the more substrate that's available to react with, the more product you'll produce. So the rate of the reaction will increase. Of course, remember that there is a maximum point at which that works, because it does still also depend on enzyme concentration. And then finally, we have the environment. So environmental factors would be things like the solvent, the pH, and temperature. Now, before we go, let's talk about enzyme inhibition because this can also affect enzyme activity. So there are molecules that can inhibit enzyme activity or even just decrease enzyme activity. And there are two types of enzyme inhibition. There's irreversible inhibition and reversible inhibition. So irreversible inhibitors covalently bond with the enzyme and they actually make the enzyme inactive. So these would be things like poisons or even antibiotics, for example. So sometimes it's a positive, sometimes it's a negative. So for instance, many poisons are irreversible inhibitors. So this would be something like cyanide, mercury, or lead. So if you ingest too much mercury or lead or cyanide, as an example, this can cause your proteins or enzymes to stop functioning. And eventually your organs will stop functioning and this can lead to fatal consequences. However, in some instances, irreversible inhibitors are a positive. So some antibiotics are irreversible inhibitors. So some examples here, we have sulfa drugs and penicillins that inhibit specific enzymes that are essential to the life of bacteria. So penicillin interferes with something called transpeptidase, which is an enzyme that's important for the construction of bacterial cell walls. But if penicillin is added to the equation, it's going to prevent those cell walls from forming, and therefore the bacteria won't survive. So again, this irreversible inhibition can be a bad thing, but it can also be a good thing, depending on the situation. So let's look a little more closely at heavy metal toxicity. So heavy metal toxicity is due to the ability to render the protein part of enzymes ineffective. So we kind of talked about this before, where, for instance, if you have a heavy metal like mercury or lead or arsenic, for instance, that can disrupt things like sulfide bonds or sulfide bridges. And this can actually cause permanent neurological damage because for instance, in your brain, there are a lot of disulfide bridges. And if you ingest mercury or lead, for instance, that can disrupt those bridges. But this can also lead to protein denaturation as well. Now, there's also reversible inhibition. So in reversible inhibition, an inhibitor will bind with an enzyme, but it can also leave the enzyme. So it's not covalently bonded necessarily. It can easily, attach or leave. So there's something called a competitive inhibitor, and this will compete with the substrate for binding at the active site. So if we look down below, we have our enzyme, and you'll notice that we've got our substrate, which fits nicely into the enzyme active site, but we also have an inhibitor that could kind of fit in there as well. So that could block the substrate from binding with the enzyme, and that could decrease enzyme activity, so decreasing the amount of product that forms. So one way that you could reverse this action is by increasing the substrate concentration, so then it overpowers the inhibitor. Non-competitive inhibitors, on the other hand, will bind to the enzyme at a location other than the active site, and that can actually sometimes change the way that the active site is shaped. So let's say we've got our enzyme here and we've got our active site, which fits the substrate perfectly, but then this inhibitor is going to bond in this spot over here. So once the inhibitor is bonded, that changes the shape of the active site, and the substrate can no longer bind to that site. So no matter how much substrate you have in this situation, uh, there's just no way that you can compete with something that changes the active site on a different location. Now, enzyme activity, like we said before, can be altered by the binding of a different molecule, so an inhibitor, but sometimes we actually call this a modulator. So modulators can influence what's called allosteric enzyme activity by either increasing it or decreasing it. So we already talked about inhibitors, which decrease enzyme activity, but there's also activators, which increase enzyme activity. Now, if you're wondering what allosteric means, down below, allosteric relates to the activity of a protein through the binding of a modulator. So a modulator affects the activity of an enzyme, and then we would call it allosteric enzyme activity. So there's something called negative feedback inhibition, and this is an example of a modulator that decreases the activity of an enzyme. So that would be an inhibitor. So let's look at an example of a negative, negative feedback inhibition. I forgot the word feedback for a second. Okay, so let's say we're trying to synthesize the amino acid isoleucine. And in order to do that, we start with the amino acid threonine. Now, in the first step, an enzyme called threonine deaminase reacts with threonine, and then there's 4 more steps that take place to get to our final product, isoleucine. So isoleucine and threonine, you can see, have very different structures. And isoleucine is actually considered a non-competitive inhibitor because it has such a different structure from threonine. Now, isoleucine is actually going to bind to a site on threonine deaminase and inhibit it. So this is kind of crazy because we're trying to produce isoleucine, but the more isoleucine that we create, the more that we're inhibiting the formation of more isoleucine. So this could be a way that you prevent too much isoleucine from forming. So your body might say, okay, we have enough isoleucine, you can stop now. Because sometimes if you have too much of something, that can create negative effects. So sometimes this is a good thing, but sometimes it can be a bad thing. So if your body does need more isoleucine, the fact that the production of isoleucine prevents more isoleucine, that could be bad, but it kind of depends on the situation. Okay, so anyway, let's just wrap up the enzyme chapter here. And this is a nice little flow chart that summarizes what we talked about. So we said that there are inhibitors, and some of them can bind tightly to an enzyme, and those are called irreversible inhibitors. So these would be things like poisons, cyanides, heavy metals, etc. We also have reversible inhibitors. So sometimes those reversible inhibitors will bind to the active site and compete with the substrate. So this would be a competitive inhibitor. But sometimes that reversible inhibitor will bind at a site other than the active site, and this would be a non-competitive inhibitor. And so we saw the example of isoleucine. All right, so that's it for the enzyme chapter. If you do have any questions, please feel free to contact me through Canvas or email, or you can set up a one-on-one appointment with me. I will see you in the next video.